Every living being is made up of very small life units, called cells. Human body is made up of more than 200 types of cells, each designated to a particular function. For example, skin cells, nerve cells, heart cells, muscle cells, etc. Stem cells are, however, different. They have capacity not only to make more stem cells by dividing through mitosis, but also to make different cell types. Practically, stem cells can be defined as cells which have potential to regenerate tissue over a lifetime. In mammals, there are two broad types of cells. Embryonic stem cells, which are isolated from the inner cell mass of blastocysts. And adult stem cells, which are found in various tissues. On the basis of potential to differentiate into different cell types. Stem cells can be of mainly 5 types. Totipotent, also called as omnipotent stem cells. These stem cells can differentiate into embryonic and extra-embryonic cell types. Such cells can construct a complete viable organism. These cells are produced from the fusion of an egg and a sperm cell. Cells produced by the first few divisions of the fertilized egg are also totipotent. Pluripotent stem cells are the descendants of totipotent cells, which can differentiate into nearly all cells, that is, cells derived from any of the 3 germ layers. Multipotent stem cells can differentiate into a number of cells, but only those of a closely related family of cells. Oligopotent stem cells can differentiate into only a few cells, such as lymphoid or myeloid stem cells. Unipotent cells can produce only one cell type, their own, but have the property of self-renewal, which distinguishes them from non-stem cells. For example, muscle stem cell. Medical researchers believe that stem cell therapy has the potential to dramatically change the treatment of human disease. A number of adult stem cell therapies already exist, particularly bone marrow transplant, that are used to treat leukemia, In the future, medical researchers anticipate being able to use technologies derived from stem cell research to treat a wider variety of diseases, including cancer, Parkinson's disease, spinal cord injuries, multiple sclerosis, and muscle damage, amongst a number of other impairments and conditions. However, there still exists a great social and scientific uncertainty surrounding stem cell research, which could possibly be overcome through public debate and future research, and further education of the public. For the purpose of treatment, these are the accessible sources of stem cells in humans. First is bone marrow, which requires extraction by harvesting, that is, drilling into bone, typically femur or iliac crest. Second, adipose tissue, or lipid cells, which requires extraction by liposuction. The third one is blood, which requires extraction through pheresis, wherein blood is drawn from the donor. Similar to blood donation, passed through a machine that extracts the stem cells and returns other portions of the blood to the donor. Apart from these, placenta and umbilical cord are also rich resources of stem cells. Banking stem cells from the placenta, in addition to those found in cord blood, significantly increases the number of parental stem cells that are collected and preserved. Stem cell treatment, however, has some concerns. For example, the risk that transplanted stem cells could form tumors or become cancerous if cell division continues uncontrollably. Stem cells are widely studied for their potential therapeutic use and for their inherent interest.